Do you have a title?
What? Yeah.
Oh, I don't see it.
Oh, I didn't put it in the... It's in there.
Okay.
It's in there. We have a title.
Oh, yeah, I see it there.
I haven't... Maybe I didn't put it in the right spot that time, but it's in there.
I see
all the things. We're doing it. Oh, we're live. Flying Out says we are live. We are here. We're here to talk about science. Boom, boom. Starting in 3, 2. This is TWiS. This Week in Science, episode number 756, recorded on Thursday, January 29th, 2020. What's up with nCoV? Hey everyone, I'm Dr. Kiki, and it is time for TWiS. Tonight we will fill your head with butterfly wings, fly brains, and a virus. But first...
Disclaimer. At the time of this disclaimer, the world is reeling from the threat of a growing pandemic, a new virus, the likes of which we've seen before, is spreading across the planet like the common cold. Scientists are watching closely, looking for the source. Is it bats, snakes, something else? Nations are taking drastic actions out of an abundance of precaution, recalling citizens from hotspots. Airlines are canceling flights. Passengers that do fly are being quarantined. Hand sanitizer sales often go hand in hand with such news, and this is a great time to be in the face mask business. And while the world braces for the worst, it is important to note that this is nothing new. Mankind has been facing novel threats from our environment since the birth of agriculture and animal domestication. Since the beginning of civilization, when we first began to live together in large groups, we exposed ourselves to an environment our evolutionary biology had not prepared us for. Ever more so in this age of technology and globalization. So, while lacking a current specific entry on the subject, the words of the standard guide to all knowledge and wisdom do come to mind. Don't panic! But maybe keep your towel over your mouth for the duration of This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. And yes, I may have said it's Thursday, but it's Wednesday, and it's Thursday somewhere in the world. We do know that's true. Oh, animals. Oh, viruses. Oh, people. The humanity of it all. We're going to be talking about all that science today. I have stories for us to keep us busy about pride, prejudice, and pheromones.
The big three, I suppose.
A little Jane Austen for you this evening on TWiS. Snakes! I did bring snakes this week, and flies. Because why not?
Why not?
What do you have, Justin?
I have got a coronavirus update. I've got the honey badger bacteria, and oh no, we're melting, as well as a fun fact about what the most complicated or complex composer might have been.
Oh my. All right, Blair, what is in the animal corner this evening?
I have some fashion-forward lizards. I have butterfly wings, and I have very smart wasps.
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 Who says a wasp?
Wasps.
Wasps. Yes. It's a very breathy wasp there.
I'm feeling very breathy today.
Oh my. Well, let's keep breathing. because it means you're alive. And let's get into the show. But let me take a couple of breaths and tell you that if you are not yet subscribed to This Week in Science, you can be. That's right. You can go to twist.org, find links for subscribing on our website, or you can look for This Week in Science in all the major podcast directories. You can also find This Week in Science on Facebook and YouTube. It's very easy. Go look for us. Please subscribe and tell a friend. Okay, let's jump in. And I think we're going to start the show with a bit of an update. We got some comments on our social media this week because of the snake virus story that was discussed last week. Oh, but in the beginnings of a viral outbreak, things move quickly. New data is being assimilated 24 hours a day around the globe by scientists, by countries, and things get changed and added up. And Justin, what happened to that snake story?
It's still live. It's still out there. It has what's basically come about is there have been other scientists have weighed in who are doubting the efficacy of this study. The reason being, just because they have found similarities to what they think would have come from originally a bat. The original story says it comes from a bat, combines with something else, and is likely then in snakes before being transmitted to humans. There's other people who are doubting that, seeing as how most this virus really comes from bats. The SARS was from bats. Even the MERS that's transmitted by camels started in bats. So they're saying it's more likely that there's a mammal if it's not directly from bats. What they're suggesting, too, is the way that we could actually determine if it was from snakes would be to see if snakes could be infected by this virus, something that wasn't done at the time of the release last week. And from my knowledge, still has not been done.
Right. What they did is they looked at the genetics of the virus, and there were some similarities and some markers that suggested snakes. And they still, like you said, they haven't 100% pinned it on bats yet. There's like a 98% probability, which is pretty close that it is from bats, but it's not 100%. I mean,
right,
oh, go ahead.
Oh yeah, well, part of it is because it is essentially a novel virus. We haven't seen it before. They've been comparing it to what they have seen elsewhere in nature and the locations where this virus has been, similar viruses have been found. And yeah, heavy hit for bats, as well as some unknown. But yeah, until they actually find it directly from a source animal, it is still to be updated.
Right. So source yet to be determined, but we do know that it is spreading around the globe. So it is something that is, like you said, novel. This 2019 nCoV is a coronavirus. Viruses are actually very common and infect people regularly. The bad ones like SARS and MERS, we don't have the issue comes that we don't have any ability to fight it off. We haven't seen them before. So like this new one, nCoV, our bodies have never seen it. They don't recognize it, and so the immune system is not ready to fight against it. But it's spreading around the globe, and the number of cases are up around 6,000 at this point that have been reported. There are 9,000 suspected further cases in China. We have still in the tens of cases, like up in the 60, 70 cases globally. But we're seeing human to human transmission, which is an issue. We still don't know how long the incubation period is.
So we do have some estimates. So I have some fun numbers here.
Okay, great. I love numbers.
So at the time of the broadcast, these are numbers. These are fresh off the press. There are 7,783 confirmed cases of infection with 170 deaths. Now that's 10 times the number we were talking about a week ago for both. We also know that the awareness and the investigation had just begun at that time. So the additional cases, these thousands more cases, are not necessarily new cases in terms of having been
they're just
contracted.
identified. Yeah.
They're identified. So the incubation time by the CDC is estimating, it's a broad range still, so it's not totally nailed down, but they've got it between 2 to 14 days. So even the reaction now, highly successful quarantining, getting precautions in place, we may still have many more cases incubating that we haven't caught yet. In terms of mortality, it's still consistent with what we reported last week at about 2%. The official estimates are between 2 and 4%. There is the rate is subject somewhat to where you happen to be. Which might be healthcare availability, might be a big factor here, or being aware that you've got this. Okay, so if you look at those cases that are outside of China, the rate is 0.3%, the fatality rate. So 0.3%, it's kind of in there with a regular flu. So regular flu is considered 0.1% in the United States, which has really good healthcare. However, if you're over 65, if you're vulnerable immune system, it goes up to 1%. So it can actually be even higher depending on which group you're talking about. So individuals diagnosed in outside of China, 0.3%. In China, but not in the hotspot, it's 1%. Still pretty consistent with a regular flu. If you take in other factors, it's as high as 5.5% in the city of Wuhan. Now, this is also where the epicenter of the outbreak was. The awareness wasn't maybe there at first, and you have to wonder: is this healthcare issue? Who's getting infected? Is it workers at these markets? These are animal food markets. They're probably not covered by a traditional HMO or just guessing. So. In contrast, the last SARS virus had a mortality rate of 9.6%. That's pretty decent, right? That's up there. According to, this is also CDC. The 5 cases that are outside that are in the United States currently, there's 0 fatalities. I wonder if back up and get that real quick. So last SARS virus mortality rate of 9.6%. To add some more comparables, the Middle East respiratory syndrome, that's the MERS that was from bats, then went into camels, and then went into people who were very close with the camels. That had a fatality rate of over 34%. And in H5N1, the bird flu, avian flu, it was 60%. Something interesting to keep in mind there in the last SARS outbreak, there were at 9.6% mortality rate, there were a little over 8,000 people who were infected by the end of the outbreak, by the time we got containment. We're already much higher than this. Or we're almost going to hit that number. I'm sorry, we're below it still, 7,800 something. We're still below it. However, if you track it, it also kind of goes with fatality. In the MERS, it was 2,400 cases at 34% fatality rate, 60% fatality rate. In the H5N1, there was only 700 cases of people dying from this disease. So when you look at why this took off, it's because it's probably indistinguishable from the regular flu.
Absolutely, and that's something that has the same symptoms, and that's something that the CDC and the World Health Organization are recommending, is that if you have not yet been vaccinated for the flu, it is still flu season. If you were to get yourself vaccinated, it would help to reduce the load on the healthcare system in terms of recognizing what they're dealing with. It just limits the number of questions that have to be asked.
I also heard they're trying to build a new hospital in Wuhan in 10 days.
They are, yes.
Which yes. You could probably build anything in 10 days. Right. It's just how well is it going to
exactly. Yeah. But it's pretty amazing the response. I'm pretty impressed by it. The fact that they're
They are sharing very well their good responses, but they are hiding some of the political aspects that have caused problems in their sharing of information at the early stages of this outbreak. So,
And that's a very good point.
That was
We
part of the
could have
problem
no-
with SARS, right? Is
Yeah.
that like they didn't fully report right away, and so it spread way faster than it should have. So they're trying to cover themselves here
Yeah,
and
and
make
I think-
sure.
Yeah, I
everything
think
is
what
done right.
happened is it potentially had a similar start, but then other people stepped in and were able to get the word out so that the international community could actually get a handle on this. I mean, I think with all these numbers and the comparisons, it is incredibly important to remember that we're at the very early stages of whatever this is, and it could end up being something that is similar to the flu in terms of severity. The flu, we don't think about it that much. People who get the flu, you maybe get vaccinated. We have a vaccine for the flu. We don't have a vaccine for this coronavirus yet, although they're working out the genetics and they're going to try and figure it out. It does appear to cause pneumonia and lead to issues in older people. So this is something that whereas like SARS was hitting young people, so people in the middle of their life more than older people,
And MERS gets anybody.
And MERS gets anybody, yeah. But the thing is, we are still collecting data. This is brand new. We know what coronaviruses are. We know what they look like. We know how they're made up. There are these things called spike proteins that are on the outside of the capsid of the virus. And these spike proteins, once you know them and they have the genetic sequence and they've already started, culturing this virus in the lab, they're going to be able to determine how to target it, it's probably going to happen very quickly. This is already a much faster response, and they're collecting data, and the epidemiologists are already working on this so much faster than we did for SARS. So 20 years ago is a completely different story from today. And I think it's very exciting in terms of our ability to catch it and to move quickly. I think we are benefiting from that. However, it's still scary, and just know that
don't panic. It's
don't
not
panic,
that scary.
though.
It's really
Yeah.
this
Don't
is
panic.
I mean,
Wash your hands.
But what I mean, what
call a
I meant to
doctor.
say is that it's scary. The headlines can be scary.
Yeah.
The social media can be scary. There are groups online who are telling people to drink bleach if you think that you have the coronavirus.
No, yeah.
Do not do that.
No,
Right?
don't do
There
that.
is a lot of misinformation being handed out as a part of this. And so really be on the lookout. This is the situation where you have trusted sources that you go to be able to get this information. I've been going to Johns Hopkins University. It's one of the on the screen right now is a map of the outbreak. This is data that's being visualized by a team at John Hopkins University. They also have a daily email newsletter that's going out updating about various aspects of what's happening. The CDC is also working on this. And then there are several really good sources online, but they have viral epidemiologist somewhere in their title. They work with viruses, they understand viruses, and those are the people that you want to be trusting and talking to.
Yeah. So here's, and then I have a quick take on it. So we've got the map up there. There's going to be a link to our website on our website with that map. So if, even though you're not panicking, but you want to panic a little bit and watch the numbers and the places where it's popping up as this thing continues to grow, you can go click on that and watch this. If you had seen this map though of a regular virus, I don't know if you can move it over to the United States. There's just a couple of little dots there. But if you moved it over to the United States and you were looking at the seasonal flu, the entire United States would be dark red. with a little bit of pink in Hawaii, but it would be dark red. the death count, it would be about 35,500, and the total confirmed would be in the tens of millions. So and the death rate. So there is, I think, an overreaction based on how fatal this thing is. However, this is a fantastic exercise in containment and analysis and reaction. And I'm all for shutting down airports, shutting down businesses, everything that they need to do, because while this one does not appear to be extremely lethal, this is the one you want to learn from. This is the one you want to practice on.
Yeah. This is our practice round.
This is the one
The drill
that
vaccine.
if we can learn from this and get this right, this greatly reduces what could happen next time, which we already kind of ran into with the avian flu and the original SARS. We've had a couple of really nasty test runs and MERS as well. But this one is the better case scenario for attempting this on a global scale to attack a virus. And look at this too. This is a virus that has circumvented, smaller viruses, this circumvented the globe. Scariest thing on this, scariest thing on this map though, the scary thing isn't the total confirmed. It's not the death count. If you go up to China, you go back to China and on this map, and you can see there's China, there's Japan, there's South Korea. One place that doesn't have any reported cases is Russia, which is a big trading partner, which does have lots of interactions with China.
So reported is the key word.
Exactly.
Perhaps.
So one of the things that I'm seeing from this map is in the quiet areas that have interplay and interconnection with China that are this silent, means that not that they haven't reacted, not that they haven't identified or confirmed or aren't having a response, but they're not reporting the data. So these are blind spots that if this wasn't the test run, as I'm calling it, for a really bad pandemic, these are blind spots that we need to collect. I mean, the fact that there's nothing really going on in all of India, nothing going on in China, these are red flags.
Yeah, absolutely red flags. I mean, we really are only as good as the data that we have. Yeah. Transparency means everything in a situation like this. Yeah. But as we have said, at this point in time, the best thing you can do is whatever is the thing that you normally do to try not to catch a cold, to try not to catch a flu. Wash your hands, avoid touching lots of things when you're in public, and also avoid touching your face. Keep your hands away from your face if you can. I know this is very hard with small children. And if you're in an airport,
bring your Purell.
Bring Purell. Wash your hands, use the hand sanitizer. These are tried and true tactics. And, if you are in public and you're going on an airplane and you don't feel comfortable, get a surgical mask and wear a surgical mask. It might help to stop from the inhaling of any airborne viruses or bacteria. But that in general.
For the most part, my understanding is the mask mostly limits your ability to transmit to somebody else.
Mostly,
it's your touch. It might be airborne, but it's gonna be your fingers mostly that are gonna get you sick. Yeah. Covering your breath might prevent other people when you sneeze if you've got it.
Yeah, what is it called? The vampire sneeze or cough?
Oh yes.
Put your cough into your elbow.
I'm familiar.
Yes, make yourself look like a vampire, like Dracula. Yeah, these are the common things, and that's what you can do. And if you are sick, stay home. Don't go on that trip. Maybe don't be a part of spreading diseases. It's hard to do. I know.
Well, that's the thing, because if it's a 2-week incubation, you want everybody wearing a mask so they're not transmitting it, because they don't even know they've got it. They're not feeling symptoms. They're not even feeling sick for weeks.
Maybe. Yeah. This is something we still don't know either. The incubation period, is it 2 weeks? Is it 5 days? And
2 to 14 is
that's a pretty big span.
2
Yeah,
days,
but
2
it's
days
based
to
off
14
of-
days. Yeah.
I'm sure they're not just picking numbers out of a hat. I'm sure this is based on when somebody was in the area of the epicenter, and then when they got
Yeah.
sick, and how these. Yes.
More
I mean,
data,
the flu
it'll
is like
get more
2 to
accurate.
7 or something like that. So
Yeah.
I feel like, it's not that far off.
Yep. Okay. Well, it's a new thing. It's kind of, it's interesting to watch, and see what's happening and the response. Don't freak out. Protect yourself. And let's move on to some other stories. There's some real cool science out there. Yeah?
Yeah, what do you got, Kiki?
What do I have? I have a story about snakes.
Yes.
Yes. Well, we've talked on the show a lot about. I brought this story to the show tonight because of the snake connection to the bats and all that kind of stuff. But it has absolutely nothing to do with coronavirus or anything of the sort. We have talked on the show previously about snake venom and how difficult it is to create anti-venom for snake bites. Right? We don't actually have a lot of snake venom with which to figure out what the anti-venom should be to be able to produce an anti-venom. There are people their job
milking snakes.
Yep.
Yeah.
Snake venom milkers.
Yep.
That's right.
Which is exactly what it sounds like. You grab a venomous snake and you make it angry to the point where it lunges, and you have a nice vial with a kind of like a rubber or latex lid on the top. So then they puncture that as if it was skin on an animal, and the venom flows through the fangs into the vial. So that's somebody's job. And then they put the snake back.
Well, maybe some graduate students have reduced the need for people to have jobs as snake milkers. Possibly. We'll see. But some researchers from Hubrecht Institute for Developmental Biology and Stem Cell Research published a paper in Cell this last week about organoids that they made for snake venom glands.
What?
They got this idea in their head. These graduate students, they wondered if they could grow organoids from other species. They had protocols in place to grow organoids, brain organoids, other things from human and mice cells, but they hadn't really looked at anything else. And so they decided they got an egg from a, excuse me. Leftovers from my winter cold. Hello.
Did we all? I just sneezed too. Do we all have it?
We all have it. My
This
goodness.
is getting serious.
Anyway, serious things. Okay, back to the story. The
So eggs.
eggs the grad students received the egg of a Cape coral cobra. And they used the protocols for the mammalian cells to create mini venom glands.
Mini venom glands.
I apologize. Hello. Yes, and these mini venom glands produce the same toxins as real snake venom glands. And so they got eggs from other species, and they grew all sorts of venom glands from other venomous snake species. Of course, they had to keep the cell culture a little bit cooler than mice and human cells, because why, Blair?
Because reptiles are ectothermic.
Yes, that's exactly right. They're cold-blooded.
Yes, I did it. Except
You
their blood's
did.
not actually cold. It's just the same temperature as it is outside.
Well, they don't need to be as warm. Their body
Yeah.
temperatures generally are a little cooler than the
More fluid. They have a larger
mammalian.
range of acceptable temperatures. Yeah.
Yes.
Yes.
And so they have created these organoids for snake venom glands. And what this potentially does is not only opens up their ability to be able to study snake venom, but the cell biology of the gland itself is going to be easier to understand. And if you can create a whole bunch of snake venom glands in the laboratory, it's going to make it a lot easier to mass produce antivenom.
So the really interesting thing about antivenom too is that you can make one and it'll work okay for several species or subspecies, but if you can specialize the antivenom to the species, or more specifically to the subspecies of snake, it can act faster and more effectively. You can lose less tissue. There's all sorts of things that are better off about it. So when I was working at the Jerusalem Zoo, for example, and they had a bunch of venomous snakes there, they had a bunch of different subspecies of Palestine vipers, and they are some of the most venomous snakes on the planet. And they were able to identify these subspecies and have region-specific antivenoms. So depending on where specifically you were bit, was it in this valley or that valley,
Wow.
you would receive a different antivenom because it is so specific.
Interesting.
So this is a situation where if you could, without having to milk every subspecies or geographical variation
Yeah.
of a snake, without having to do that, by just using the genetics of that animal and being able to make a species-specific and region-specific antivenom, you could save lives and you could way reduce the cost and the effort it takes to do this. Not to mention the people milking snakes and the snakes being milked probably would be a lot would be less stressed out in general. It would be good. I think those people wouldn't mind potentially
wouldn't
being out of
mind
a
it.
job.
Yeah.
Yeah.
They may enjoy doing something else.
Yeah, I mean, even
Spending
if
their
they
time
enjoy
doing
doing
something
it,
else.
I would say a lot of those people have at least had close calls, if not ended up in the hospital. So,
Right. And
it
the
would
snakes aren't,
be-
it doesn't make the snakes happy either.
No,
It's not.
and it's
not gonna be fun
important.
for the
Like I'm
snakes.
not, I don't want to open up a whole conversation about, oh, those poor snakes. Yes, those poor snakes. But just like those poor mice that have helped develop potential cancer cures, where there's a trade-off here, and a little bit of discomfort from these snakes. If it can save a bunch of human lives by developing antivenoms, I'm not going to get
You're
too far
in.
into it. I'm into it. I'm into it.
You're in.
Because also there's this whole issue with like people hating snakes and snakes having really important ecological roles, and maybe the hatred and fear will be a little bit less if there's less imminent danger, which, you know, modern medicine and antivenoms help with that as well.
Yeah.
Whole thing.
It's a whole thing. Exactly. Make them, they could be all better.
Mm-hmm.
So moving on from snakes and venom. Venom, not poison. Remember, venom is injected. Poison you drink.
Ingested.
Ingested. Yes.
Injected, ingested.
Flies. They're brains. They're tiny.
Truth.
And so researchers, it's true, yes.
That's a true fact.
Nobody's going to argue with you.
Yeah.
So researchers had at a research institution called Janelia, this is a Howard Hughes Medical Institute, Janelia Research Campus, have published in the bioarchive their first ever fly brain connectome map. Now, this is a big deal because the last connectome, and what this is the neurons in the brain and how they connect to each other.
It's basically a neural network, but it's so small and it's just a little ball of things that it's a little more
not
simple.
Right. So if you were thinking about the human brain, it's like, oh, it's not that ball of pink jello. It's the neurons within it, and so fine scaled and high resolution that you know how all the neurons in it are connecting to each other as well. Anyway, we haven't done that with the human brain. We are very, very far away from doing that with the human brain. The last organism that had a connectome map made for it was about 1989, and it was the sea slug. Very small brain, something like 7 neurons, and maybe 1,000 or so connections. Very small map, very, very small. Now, this Yanelia brain is a hemibrain. It's about 1/3 of the actual size of the fruit fly's brain, the Drosophila melanogaster, and in it they have mapped 25,000 neurons and about 2 million connections. And they have been able to identify over 4,000 distinct kinds of neurons among those 25,000. Now, this, I think, is one of the big indicators of progress in terms of understanding the brain, is this ability to be able to identify not just, oh, there's a bunch of neurons connecting to each other in the brain, but what they are, what they do, what kind of groups of these powerful cells work together to give us the structures in the brain and allow us to do the behaviors that we do. This hemibrain connectome took 12 years and $40 million to complete, and a lot of technology that didn't exist 20, 30 years ago.
I mean, it wasn't that long, maybe 15 years ago, my invertebrate biology college professor told me insect brains, they're pretty simple. They're basically just a ball of nerves. It's like a wad. Like, it's simple. There's not a whole lot going on in there. It's just a wad of nerves with a very little order.
And what do you think now, Blair?
Shock and awe. That animal's smarter than we thought. It's like the moral of most animal intelligence stories on this show. Oh my, they're smarter than we thought.
Yeah.
So
Now,
it's one of those interesting things. Like, I wonder how much like that's a pretty amazing map of connectivity there. Takes a lot just to be a fly. Don't underestimate it. But I wonder how I want to see the comparative, like human versus chimp or something along the lines of where does the difference of connectivity in this sentient hairless ape brain with the rest of the brains out there?
Right. So what kinds of neurons are there? So, those 4,000 distinct types out of the 25,000 in the fly brain, how many kinds do we have in our brain versus the primate brain, right? And how do they stack up against each other in different parts of the brain? Do you have a higher proportion of one kind of neuron than another in the human brain versus the primate brain? That is something absolutely we need to know that would tell us a lot about our evolution. Even starting with the fly brain and this small portion of it, this is going to tell a lot about just the structure that leads to the function of the brain, of the behavior of the animal. And the way that they did it is actually really, it's the technology is fascinating because, okay, it's a brain, it's a fly brain, it's tiny. They sliced it up into little tiny slices.
Can't even imagine.
Right, they did their tissue preparation to get it ready for slicing and staining. They stained it so they could see the neurons, and then they took pictures of it. Those slices got imaged, those scans were put in a computer, and then this is where the fun part happens. This is where they start adding in algorithms to put the puzzle back together, because you have the slices through space of the brain, and a neuron, you can have a dendrite tip in one part of the brain that leads back to a cell body all the way back in the other end of the brain, right? Through that one neuron that you can follow through several consecutive segments of the brain. And to do that, there's actually, you can hand draw it and try and figure out how it all links together, but they have developed imaging techniques and algorithms that go pixel by pixel to paint in the neurons and to determine their three-dimensional structure. And so that in itself is really exciting. And now that we have these new technologies, perhaps they will help things speed up. And so I couldn't help but think at this 12-year number, I was like, oh, that's like how long it took to get the first genetic sequence. Right? It was like 12 or 13 years for us to go from the beginning of the Human Genome Project to we have a full, complete human genome. And then over the following decade, more and more genomes and sequencing got faster and faster, and it's gotten cheaper and cheaper. And yeah, we might still be trying to figure out what to do with all of that, that data and information and what does it mean. But we've gotten faster and better at compiling it. And so I can only imagine that this step is going to help other efforts that are taking place.
Yeah, Human Genome Project was like 10 years, $1 billion. On a nanopore sequencer, I think you can get a human genome and basically over 24 hours and a few $100.
Yeah. Pretty exciting stuff. So anyway, fly brains, they excite me. Woohoo! Who knew? I didn't know I would be so excited by a fly brain. I didn't. But anyway, there we will put the links to all of this. It's bioRxiv, which means it hasn't been peer reviewed yet, but it is publicly available. So there's access to all of these things, and we will have links on our website, twis.org. Oh, is it getting to that time in the show?
I think it is.
There's this time.
Send Blair to the corner.
Oh, time to send Blair to the corner.
My corner!
Yay!
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's animal corner.
With Blair. What you got, Blair?
Well, Kiki was just talking about fly brains. I want to talk about butterfly wings. Butterfly wings. What are butterfly wings for? What do you think?
It's
Butterfly
flying
in the sky.
Oh,
I can fly twice as high. There we go. Yeah.
take a look. It's in a book.
In a book. Reading. Oh, anyway. Yes. Butterfly.
Butterfly wings. They're for flying. This is a study from Columbia Engineering and Harvard. And the understanding until this new study is that butterfly wings are pretty lifeless membranes. They're just appendages that they use to fly. But this new study shows that butterfly wings actually have a network of living cells whose function requires a constrained range of temperatures for optimal performance. So first of all, that it's kind of like living and moving and breathing in its own way, but also that it has these wings have very specific temperature ranges that they need to be able to make butterflies fly. So by carefully removing wing scales, they were able to peer into the interior of wings. They stained the neurons within the wings and found that butterfly wings are loaded with mechanical and temperature sensors. So there is a lot going on through these butterfly wings.
Wait. They've got mechanoreceptors and temperature receptors in their wings?
Correct.
Wow.
So
Sadie's excited by that also.
Well, and what a wonderful idea for you've got these giant sensors out there, these panels that can be collecting data and informing the little wad of nerves or the very complex brain as it might be.
Yeah,
of a
absolutely. And so they're calling this the wing heart. It beats a few dozen times per minute, and it helps facilitate directional flow of insect blood or hemolymph through a scent pad, as they have decided to call it, which is an organ on the wings of some species of butterflies. They can actually look at the butterfly wings now as dynamic living structures. They're not just an inert membrane. And this is what really got me interested, is that the new hypothesis now, we've only ever looked at butterfly wings as functional things that help them fly, and the other thing that they do is help them display specific colors and patterns for attracting each other, or more often either camouflaging or creating scary or misleading markings to kind of scare away predators. The new theory is that patterns on their wings might also help them to modulate temperatures in the living parts of their wings. So they might have dark or light parts of their wings, like in all of the zebra stripe stories we've talked about, how that creates kind of like a convection current or helps heat up or cool down different areas. This could actually help with that as well. So this, is a whole new thing.
So if you could reverse engineer like where the nerve networks are and what their functions are, and see some sort of pattern
to the coloration and the structure of the wings,
be interesting.
Sadie's so excited about it, so bargain over there. They also, the next step they did is they did infrared hyperspectral imaging, so that they could look at the temperature distributions over the butterfly wings, which helped them kind of see how these different patterns could relate to that, but also see that there is actual flow of temperature across the wings that is not just environmental. It is facilitating this in some way. And so they then took experimental conditions to mimic butterflies' natural environments, and they looked at relative contributions of environmental factors to wing temperature, including intensity of sunlight, temperature of their terrestrial environment where the butterflies are standing or flying over, and the coldness of the sky. So apparently, when they're remaining dormant, just kind of sitting, they have the potential for their wings to overheat. When they're flying, they have the potential for their wings to get too cold and kind of freeze, which was kind of the opposite of what I expected. Just because the way our muscles work, when we're moving around, we heat up, but that's again because we're warm-blooded, so there's a whole nother thing going on. But with these guys, because they're flapping through the quote-unquote coldness of the air, it way dips the temperature of the wings. So there's all these different factors going into the temperature of their wings. And so the team found that in the simulated environmental conditions, despite diverse colors and patterns, the areas of butterfly wings that have live cells, so they have wing veins and scent pads, are always cooler than the lifeless regions of the wings. So they have radiative cooling. So these nanostructures in the wing scales could inspire radiative cooling materials to help with excessive heat conditions and other things that we do, like, for example, in flying robots. That would be a thing that we could do.
Of course, it always comes back to flying robots. It always
comes back to flying robots, of course. So they found each wing of a butterfly has about a few dozen mechanical sensors that give real-time feedback to enable complex flying patterns. And that also, too, could help be inspiration for flying robots. So basically, all of this comes down to, how can we make a better flying robot? But also, here's another kind of simple invertebrate that we kind of thought we understood. They just had wings. They were just like keratinous membranes that they use to fly. But there's so much more going on there, which also, by the way, I cannot bring up butterflies and the complexity of their wings without mentioning the fact that when a caterpillar turns into a butterfly, it's basically muck in the chrysalis. It is not very organized. I mean, more science that we're getting on this shows that there might be some sort of body organization still going on in there, but for the most part, this caterpillar liquefies in there. There's very little order. And so the fact that caterpillar muck can turn into this is just so cool.
Wait a sec. So do they not know how the muck turns into a butterfly?
Not completely.
Yeah,
no, we're
really.
still
Oh my
figuring it
goodness.
out. Yeah.
That sounds like a fun thing to investigate. Because there's got to be
a lot of research going on with it. Yeah.
there's got to be a starting point to get something this organized.
Yeah. I mean, there's a starting point to getting it so disorganized, and then there's a starting point in getting it reorganized, which is all, I think, fascinating.
Yeah.
It really is. And that such beauty comes out of it. And not only that, but really these functional wings. This is
They have their own heartbeat.
They do. I am even more impressed with butterflies, and I from now on do an even better job of keeping children away from butterfly wings.
Oh gosh, for sure. So next, I have to tell you, I know it's early in 2020, but I'm going to go ahead and tell you this is my favorite story of 2020 so far.
Oh, okay. All right, bring it.
The color of your clothing can impact the behavior of wildlife. This is a study from Binghamton University in New York. So they were looking at water anoles, Anolis aquaticus, which is a lizard in Costa Rica. They were chosen as a study subject because they have a small range. They don't move very far outside of their home range, and they are very remote. So they haven't seen many, if any, humans in their lifetimes if you go to Costa Rica and look for water anoles. So that means that before you go in there, if you're doing any experimentation in relation to humans, you can have good confidence that these populations were not biased by previous human interaction. That's important because this study wanted to see how human clothing could impact wildlife. They visited 3 different river locations wearing one of 3 different colored shirts: orange, green, or blue. The idea was to see how the color that you are wearing could impact how many anoles you see and their behavior. Orange was chosen because the water anole has orange sexual signals. So that's their kind of attention-getting color. Blue was a contrast to orange. For those of us with complete color vision, we know that orange and blue are very different. And green was chosen because the tropical forest is green. So they thought it would be a good camouflage color for the humans. So the idea is, do they want to see orange, humans wearing orange? Does that make them more comfortable because they're used to that signal? Do they want to see blue because it's unlike an anole, and so it's very clearly labeling a human as other? Or are they more comfortable when they can't see the humans at all, perhaps because they're blending in with the environment well? So the hypothesis was that colors worn by anoles, the orange, would be less frightening because it's a familiar cue. That was the expectation. And in fact, that hypothesis proved true. Researchers with orange shirts reported seeing more anoles per hour and had a higher anole capture percentage for when they were doing surveys. And despite predicting that, the lead researcher was still surprised by the findings. She said, quote, "It was still very surprising to see that the color green, which camouflaged us well in the forest, was less effective than wearing bright orange." So this is a good reminder that animals perceive the world differently than we do, or perhaps the same, but that ultimately our behavior in animals' space has an impact. Even if it's deciding what shirt you're going to wear that day, that could be an impact. So this, not to have another story that's like throw out all the data. But if you're doing wildlife surveying and you think you have specific ideas of seasonal habits or anything like that based on your surveys, if humans weren't around, maybe your winter coat was distracting. And that's why you saw more animals then. You know, there could be all sorts of stuff related to how we present ourselves in their space that could be a confounding variable.
Oh my goodness, this is just human centric. You know, it's you've brought me a new perspective that I hadn't considered, but it is so true. It is their space. I mean, nature is our space too, but more often than not, if we're just going there to observe, to do studies or go hiking, you don't, I mean, yeah, animals can see you too.
Animals can see you.
And they are going to respond to that in different ways. Yeah, this is a very interesting idea that needs a little bit more.
A little bit more. I'm dubious. Count me super dubious.
I mean, I definitely know, for example, there are animals I have worked with, domestic and wild, that have responded to me differently when I'm wearing a hat. We know this. We know that there are visual cues that my dog is scared of children but only when they're wearing hoods, which I think is so funny. Like there's all sorts of weird cues that we give animals that we don't realize. I mean, this makes nothing but sense to me.
Animals are not people. They do not see the world the way we do.
They're not just like, oh, that's a cool shirt. No, it's like, oh, what is that color? Is that a warning signal? What's happening?
That I think is really one of the points. Is it a warning signal? Is it something I need to be concerned about? Is there danger?
But this isn't it? I don't know. I'm still going to remain dubious on this for a bit.
Yeah,
but,
sure.
But it's like for the longest time, bulls and the color red.
Right.
Ooh, the color red would get the bull going. And then it turned out you could actually flap any type of fabric around a bull, and they'd be like upset by it. It
sure, but also when
that is more for the audience at that point. So
when it comes to
doing a bullfight or something.
Yes. When it comes to animal color vision, there's still a lot of question marks.
Yeah, so this also makes a lot of presumptions about the vision capabilities or how they're not. I mean, if anything, if I were to guess the story being more, I'd make it even more human-centric and say, the humans who are wearing orange were more tuned to seeing orange in their environment. Something of this nature.
Interesting.
I'd go the other way. I have a hard time feeling like the Knowles really cared that much about it.
Well, the thing you should do in this case is have the camera trap be different colors.
Yes.
Oh, perfect. Yeah, there you go.
So you take the human element out of the equation there.
And repeat it over a very large set so that you didn't just go in and out. This
is a preliminary study, but it looks fun. It looks like it's something that we might need to start considering, which has a couple implications.
Cultural
so one is
awareness for animals.
Yeah. So one is when you're doing research, but the other is if you're an ecotourist. If you're going into wild places trying to see animals, it might be time for the people in charge of that space to recognize what colors might make it easier to see animals, but also what colors might be stressful for the animals to see.
Yeah. So, and my guess too, if they're all watching this, human in an orange shirt, that's obviously a human. Human dressed in camouflage might look like a moving bush. That's a freaky thing. I've never seen a bush walk before. I'm getting out of here. Like, I mean, you have to get inside the head of the Anole and try to figure out what they were thinking. It's tough.
Yeah. It's an amazing eye-opening story for me. Once again, about confounding variables.
Yeah, and the things that we don't really consider.
No, if I was getting dressed to go do some research, I would not be considering what color my shirt was in relation to the color signals that my subject species was interested in.
Right. You just get dressed.
Yeah.
I want
This
to
shirt is
quick
comfortable.
I
and
do
warm.
like Ed's idea though of, it should be, maybe field research needs to be like naked and afraid.
Yes. Although bug bites. I'm just thinking Costa
bug
Rica.
bites.
It's
Yeah.
too much. Yeah, Justin?
No, never mind.
The moment passed.
Yeah.
Oh, boy.
All right. Well, I think it's time for us to take a quick break before we head into the second half of our show. We've got more science. Justin's got melting things, and I've got, what do I have? Jane Austen. It's coming up in the second half. What does that have to do with science? You'll have to wait and see. This is This Week in Science. Stay tuned for more in just a moment.
Can't explain things you've heard more than.
Thank you for listening to This Week in Science. If you love what we do, please go to our website, TWIS.org. TWIS.org is where you can find buttons to subscribe to us on Google, in the Apple Play podcast directory, not Google Play, Apple podcast directory, or YouTube. Also, you can find show notes for all of our episodes. You can subscribe to our newsletter if you don't have pop-ups blocked, and you can help support us. We are listener supported, and this year, thanks to a grant from the Borough's Welcome Fund, we are also a little bit more supported, which we really appreciate. This is going to help us out a lot this year. But if you would like to help support us, along with the Borough's Welcome Fund, you can join our Patreon group. That's right. Click on the link for Patreon. It will take you to our Patreon page, which is patreon.com/thisweekinscience, and you can choose your level of support. You'll be charged once per month, and just think about what kind of value we bring you week after week, the shows that you hear. What do you like about what we bring you? Do you want to help bring this value to people and to help us continue to make more of it, maybe to increase the amount of it that we do? If you like what we do, please help us to continue to grow. And tell a friend. That's right. We always appreciate you sharing TWiS with a friend, because this shouldn't be a secret. Really, it's not a secret. Tell people. Tell 5 people this week. Tell them they should listen to TWiS to get their fun weekly fill of science news discussion. Okay. Oh, really? Thank you so much for your support. We could not do what we do without you.
We can't explain things you've heard more than intuition. A lot of reason shows the way.
And we are back with more This Week in Science. Oh yeah, more science for you, for your ears. And what do we have for you now? We have a This Week in Hey TWiS. That's right. I have a letter from Bert. Bert wrote a letter and he said, "Hi, Dr. Kiki. I was just listening to the latest podcast, which was very interesting as always. I definitely enjoyed the interview. I wonder if you could find an ecologist who could talk about the impact of Trump's rollback of the clean water regulations. I remember what things were like in the 1950s, and my parents were involved in the movement that led to the passing of federal regulations protecting wetlands. Now we seem to be going back to poisoning them." I will work on that, Bert. I think that sounds like a really interesting conversation to have here. Thank you for that idea. I'll work on that. Now I've said it out loud in front of everybody, so I'm going to have to stick to it. Then he goes on to say, late in the podcast, you and Blair talked about whether parrots go deaf as they get older. I live with a male blue and gold macaw who will be 25 in May. He lives in my office and can scream so loud it hurts my ears and can drive me from the room. So I also wondered about this. Then I learned from a scientist on Naked Science from the UK that parrots and possibly other birds actually regenerate the fine hairs in their ears that get damaged by loud noises. Research is going forward to determine the exact biological method for this in the hope that someday we will be able to trigger regeneration of human hearing. I hope to live to regain the hearing I had when I was a boy. All the best, Bert. Yeah. So that conversation we had, Blair, about the parrots, it seems like there is some research in that direction, which
so
is really
cool.
interesting to hear. Yeah.
Yeah.
And
Yeah,
Bert
they are loud.
They are loud. And that blue and gold macaw, you have probably at least another 60 years of that.
Yeah. Yeah. Oh my
Maybe
God.
50, 60 years of that
Yeah.
scream.
Yeah.
It's good to have parrot family.
Yeah. Bert, have you lined up the person who is next in line to receive your parrot? That's what I want to know.
Yeah, who will be that person? Yeah. Whenever you have a parrot, you have to think of where that parrot will end up in the future. But I love the idea. I think, like we talked about, I think it would be so interesting to find out. I mean, these animals, they're very social. They are loud. They live in a loud environment. Why not? Why would they have regeneration? I bet they would.
I feel like we were talking about something along these lines a little while ago about the necessity of having a pretty advanced ear in some place as noisy as a jungle, and also the brain power to pick out specific sounds. So a regenerative ear on top of that makes perfect sense.
Yeah. I wait to hear more. Bert, thank you so much for writing in. I do appreciate your letter. It's just wonderful to hear from you. Anyone who wants to write in, please send an email. My email is Kirsten, Kirsten, at thisweekinscience.com. Tell us what you think about stories in the show, if they get you thinking about your parrots or or about any other thing, let us know what you're thinking, and I'll see if I can read it in the show. And if you do have a What Has Science Done For You Lately, I will still read that as well. You can also leave us a message on our Facebook page. But now, let's jump back in. Justin, you had more science? Yeah.
I do, and it's loading now. So basically, we've been talking about these viruses and how these viruses are dominating the news with their ability to go viral in a pandemic-y way. This is a study out of Duke University. Researchers there demonstrated that at least 25% of antibiotic-resistant pathogenic bacteria found in clinical settings are capable of spreading their resistance directly to other bacteria. 25%. That's a pretty big number. This is building off they had done in a laboratory previously. I'll just read the quote. Our previous research showed that antibiotics do not affect the rate at which bacteria spread their resistance directly to their community in laboratory strains of E. coli. But they went, this is Ling-Chong Yu, professor of biomedical engineering at Duke. But we wanted to see if this is also true for clinical strains of pathogens that are actually out there in the world. And that's what they found. They found this, and it's interesting. So it's not that antibiotic overuse isn't a problem. It's that antibiotics themselves didn't change the transfer rate. It doesn't mean that because the antibiotics have been used that there's higher transfer rates of this resistance. You get the higher resistant strains or more when you've knocked out everything that isn't resistant, and then that's what you end up with. Is what's left is these ones. So it's sort of an interesting take. I think that needs to get looked at a little bit more. But it says as antibiotics grow, scientists are trying to find new antibiotics as well as figure out how to stop resistance from spreading. Since most antibiotics are coming from natural sources, there will always be a huge reservoir out there in the wild of bacteria that are already resistant to whatever we're using as an antimicrobial at any given time. And as those wild strains interact with the pathogens, that's when we are likely to see the sharing of resistance and the problems that we are running into with coming up with antibacterials. But if we can learn how this resistance mechanism actually operates, we may be able to engineer ways to avoid them in the future.
But we do know that, I mean, an increase in the misuse of antibiotics can lead to higher population percentages of antibiotic resistance within individuals. So an individual that had bacteria in bad bacteria, you take antibiotics, maybe don't finish taking the entire
Because I feel better.
right?
Why would I
feel
still be on
fine.
these?
You know,
They hurt my stomach.
meanwhile, a few of those bacteria are still alive, and they have potentially gained or they have a mutation or they gain a mutation for resistance, and then it's those bacteria that can go on to spread. So it's. I mean, I just want to make it clear that even though the use of antibiotics doesn't affect the way that it's swapped, the rate at which it's swapped by bacteria, it's still how you use antibiotics that's the source of that antibiotic resistance in the first place.
Yeah, so it's not just that there's an overuse of antibiotics that by itself isn't the whole problem. You're correct. It's the misuse. So if you didn't knock it all out, if you didn't kill everything, there's that you still have the rate of exchange going on where they can eventually become resistant. And if you didn't kill everything, those pathogens are still in our environment. Yes.
Yeah. So that misuse is still important. So if somebody tells you to take antibiotics, make sure you're taking them for a good reason.
They say take them for 10 days. Don't stop at 9.
Mm-hmm.
It's doing it wrong. I don't care if you feel better. It's not how this works, people.
If you have a flu, don't take antibiotics.
Yeah,
flu is caused by a virus. Antibiotics kill bacteria.
Don't take antibiotics unless you're prescribed them.
And fun fact, some bacteria kill viruses.
It's true.
Yeah. So,
phages are our friends.
Yeah. So you might actually make yourself more susceptible. It's possible.
It's possible. This is interesting, though. It's kind of neat this is how the bacterial populations kind of work, and it's the mechanics of the way they swap genetic material.
Yeah, and I kind of spared the analogy that was provided. It had to do with baking cookies, and resistance is basically a recipe. And as some bacteria share cookies, they also share the recipe with how to. It was a silly analogy, but the point of it is that in understanding how those recipes are transmitted, you may actually be able to create perhaps a synthetic antibiotic or just an antibiotic in a way that's already in existence, in a way that makes it less transferable over time. That's the goal of the next focus of the research.
Cool.
Yeah.
Very cool. Other new research, I mean, I'm going to get away from viruses and microbes, but still on the molecular small scale of things, I'm going to turn to our carbon emissions. It would be really, really great if we could reduce our carbon emissions into the atmosphere. Yes, we agree. That would be pretty cool. So researchers have been trying to figure out how they can take carbon and make it useful somehow so that they can take kind of stuff that would turn into carbon waste and turn it into something that can be used. Researchers developed a new technique called flash graphene. Flash. Anyway, they
Graphene of the universe.
That's right. And in this process, they, in 10 milliseconds, flash heat carbon-containing materials. And when I say carbon-containing, I mean anything. It could be rubber tires that are carbon-based. It could be food waste. It could be, I don't know, tree stumps. It could be all sorts of things. Anything that contains carbon, it can be flash heated up to 3,000 Kelvin. And this isn't about Karen's friend Kevin. No, this is 3,000 Kelvin as in 5,000 degrees Fahrenheit. And in doing that, it breaks up the carbon so that the bonds are broken. And then as it cools from that high temperature, it recombines into the form of carbon known as graphene. Now, making graphene is very difficult and very expensive. And we use it for lots of little tiny uses currently, but it would be amazing if we could create it in bulk. And so these researchers from Rice University, based on this method, they've actually created a company. They have started a company so that they can commercialize this process. And they're thinking that they can take all sorts of waste materials and make bulk graphene. They've shown that they can take even just a very small amount of graphene, add it to concrete. It makes the concrete stronger and reduces the emissions from the production process significantly. So the idea, they say, by doing this, they're trapping greenhouse gases like carbon dioxide and methane that waste food would have emitted in the landfills. We're converting those carbons into graphene and adding that graphene to concrete, thereby lowering the amount of carbon dioxide generated in the concrete manufacture. It's a win-win environmental scenario using graphene.
Nice.
Yeah, I think it's a fairly exciting new method. I mean, of course, there is going to be, I imagine there's a massive amount of energy going in to the flash graphene process.
Yes, where's that coming from? Is that all solar powered?
Yeah, so can this be sustainably, renewably powered? Where is the energy going to come from to start the process? But if this can also, there are comments that this could also be used to change coal into graphene so that we can use waste coal instead of it being burned, that it can be turned into graphene also.
Waste
anyway,
coal?
Yeah, I don't know. I have questions about all of the things they're going to be using this for. Yeah, but it's an interesting idea, and they hope to be able to, within a year. Right now they aren't making very much of this graphene, but they're hoping to be able to produce 1 kilogram of graphene a day within 2 years. They've been funded by the Department of Energy to, and here it is, convert U.S. sourced coal into graphene. They could provide an outlet for coal in large scale by converting it inexpensively into a much higher value building material.
Wow.
Is this because there are currently coal mines, and so this is a way to keep them functioning without burning the coal? Is that what this is?
I don't know. I need to find out more about this part.
Yeah. So I kind of doubt it, Blair, that's the sole reason that the researchers involved in this project went about tackling this issue.
No, that's not.
No, I'm just saying the mention of coal specifically.
No,
there's lots of other things going on, but
yeah, but so this is also an interesting thing that I've thought of a bunch of times. Carbon is a great building block for all sorts of things, and at some point we are going to find a higher and better use for oil, okay, than plastic and just igniting it in a combustion engine. Someday in the future, you know what we could have done with all of that oil?
Too bad,
and it's amazing. Right. Hopefully it won't all be gone, but we may find a much higher, better use. This could be one of those things where hey, here's a higher, better use for the coal instead of just burning it. There's all of these other applications it could be utilized for, and there are, I mean, certainly, if we were in a post-oil world, we wouldn't be able to have plastics invented if they were, if this was years ago. So these sorts of things that we hadn't discovered. So there's could be a
But
better
now we have
use
silicon. We can just make everything out of silicon. It's fine.
Okay.
That's not going to happen. No.
Anyway, I
Carbon's
am
great.
actually suddenly reminded, not to go too far off on a tangent, that in my childhood, the big concern about oil and gas was that we were gonna run out.
Oh yeah.
There was not
oil.
a lot of conversation about climate change or the impact of using it. But it was this question that it's a finite resource. It's not renewable. So we need to come up with renewable energy plans because eventually we'll run out. Little did I know that there was something else that was going to show up that was going to be much more of a problem before we ran out.
Well,
I think there are different forces there. Yeah, but go ahead, Justin.
So it could eventually be a self-correcting problem. It's just not soon enough.
No.
So the stats on oil use is that every 10 years we use as much oil as has been used in all of the previous decades. It's going up. It's just you get the one box up top that's as big as all of the boxes below every 10 years. And so there's questions about how can we possibly continue to do this forever. Now we've become better, more efficient at using it, and things like vehicles are much more efficient than they initially were. But population grows. There's a lot more cars. Nations, entire continents that barely had cars now have cars. So that's one of the things. Then you got to look at something like Venezuela that's probably got as much oil as Saudi Arabia, but it hasn't really been gotten into that much. A lot of places in the United States actually are intentionally not drilled for oil, even though we know oil is there, like all of Southern California, because it's for a reserve if we ever needed it, kind of a thing. So the chances of hitting peak oil in time to help with global warming, not a really good option.
No,
it is a real possibility.
That it's such a different. It was still an angle of like this isn't sustainable. We have to have an exit strategy. But we were so far from the imminent concern when we were just like, oh, but it's finite. We'll run out of it.
Well, the thing is, we aren't going to necessarily run out of it. It will become financially prohibitive to continue to get it out of the ground. It's just going to, I mean, we're going into, tar sands. We are separating the oil from other substances in the earth. The procedures that are involved in a lot of the acquiring of oil become more expensive, more, just involved. And
the track of going up, a magnitude every 10 years is just, it's an insane effort.
And eventually you'll hit a price break where people won't be able to afford it anymore. And there will still be oil left, but there won't be a financial incentive. So there's the market side of it. And I'm kind of glad that climate change is getting in here and we're pushing more toward the renewables a little earlier and not waiting for that peak financial tipping point to take place.
Yeah, that would be, you're right. That would be so economically disastrous to the world to actually hit peak oil and not have a backup plan.
Yep. But speaking of climate change, Justin, didn't you have a story about something melting?
Yes, we're melting underwater, under glaciers, much quicker than previously thought, according to Rutgers' co-authored study. Findings are challenging again, as with every one of these, they've looked at a new thing. The framework for how they analyze the interactions of glaciers in the ocean, in this case, is implying that they're going much quicker. This is published in the journal Geophysical Research Letters. They surveyed the ocean in front of a 20-mile long LeConte Glacier in Alaska. This is the first time they've been able to do some of the analysis involved in this study, because this is the first time the researchers had at their disposal robotic kayaks. So they were able to put these robotic kayaks right into the plumes of meltwater that are coming off. This is water that's released when the ice is melting, right where it meets the ocean, and it's a hard place to place a probe. The probe itself might be able to survive there, but getting it to that location is exceedingly dangerous. You have the calving of ice that's falling into the ocean. You get waves from those big chunks of ice that are coming off. It's too dangerous for humans to go in there and even place floating probes. So they got these robotic kayaks. They remote controlled them in there. And this is quotey voice of lead author, who's a physical oceanographer and assistant professor, Department of Marine and Coastal Sciences in the School of Environmental and Biological Sciences at Rutgers University in Brunswick, Rebecca Jackson, a relation. With the kayaks, we found a surprising signal of melting, layers of concentrated meltwater intruding into the ocean that reveal the critical importance of a process typically neglected when modeling or estimating melt rates. Basically, you can summarize this as we're melting faster. Now, this is not the first time that we've come to this conclusion in studying glaciers. Every new look that we have made of the glaciers has given us more evidence of faster melting than the previous models. And the previous models, as they get built with new information of quicker melting, are also then updated by the new quicker model. So it's not just they're both all agreeing with each other that, yes, we're all, the other one's correct, we're this much more than the original model. Each model is finding more rapid melting than the previous model because they're looking at these glaciers from different aspects and different ways and different points of the melting taking place. So what does this mean? There is one glacier that is responsible for about 4% of sea rise that is going to be melting much quicker than predicted. So all of the models then have to take these into account. So even with the direst information you've heard from your host of This Week in Science about sea level rise and the effects and how quickly this is coming, those were underestimations.
Great.
If we work really hard, predictions won't be as bad as we think they won't come true. We have to work really hard, though.
We do. We can do that.
Yeah. So one of the things was it is in this one they're finding that the underwater mix is playing a crucial role where freshwater discharges at the base of the glacier from the upstream melt that was previously discovered. This is what's sort of driving an additional melting process that's taking place along these edges. And this is sort of the thing that hasn't been calculated into some of the other models.
Because the melting point is different from freshwater and saltwater, right?
It's the melting point's going to be different, but it's also the way it's interacting. The way it's interacting with it is more, I guess, more vigorous.
Yeah, I mean,
creating more melting.
That's going to be a fluid dynamics
A lot of
question,
fluid dynamic
right?
things. Yeah.
Yeah.
There's also these weird upwellings that take place. So we're kind of familiar when you see like a frozen pond on top of a glacier that can then just disappear. And it's disappeared because it's fallen through some cracks in the ice, and then it's the lake that was there on top of the glacier disappears. It drains through it. It can also kind of go the other way because the weight of the glacier as it's moving, if there's a lot of liquidization, it can also push liquid up into the glacier, and then you're also creating melting every time you're moving fluid water, which is obviously of a warmer temperature, in and around and through. So it's a big moving target to sort of get these estimations as it is, but the more looks that we've had at it, the faster that number of melt becomes.
And I would like to say at this time that even though it's like, oh, there's this new thing that needs to be added to the models and that needs to be considered in a new way, all of the models since the 70s have been very accurate in their predictions. We have just been fine-tuning that accuracy over the years to get rid of those error bars a little bit more. But the general trends that they have predicted since the 1970s have been, in the majority, accurate.
Well, I would disagree with you a little bit there.
There's a paper that just came out this last week. Do you remember Gavin? We interviewed him years back from he's a climate researcher at NOAA, and Gavin Schmidt, I think is his name. He and some other researchers published a paper this week that looked at at the accuracy of a bunch of models to basically ask this, get at this question of this argument against, which has been used by a lot of the debunkers who try and debunk climate change, that, the models aren't accurate. The models don't know. But the general trends we've known for over 100 years, the models have been getting better and better since the 70s, but they have, by and large, been right the whole time.
Yes, my favorite one is we've known how carbon dioxide traps heat since the Civil War.
Yeah, and so when I was disagreeing with the thing that's been changing in the models, is how quickly the prediction we're actually having to accelerate the timetable of the predictions a bit. The predictions of like how much ice is can melt and how much that will affect the sea level. These sorts of things are being pretty well tuned in, but the rate at which it's taking place is the thing that keeps getting updated. It's not that is it melting a glacier? Isn't it melting? It's definitely melting the glacier. But now we can see there's other. So because the IPCC report, there was not a lot of glacier dynamics involved in that. Most of the studies that we've talked about having to do with glacier activities, Antarctica, up in the North Pole, are pretty recent. Like I feel like it's been the last 4 or 5 years even that we've really started to get a lot of great data out of there to figure out how, as we've been getting more funding into monitoring and keeping an eye on how this is all going down. But you're right, Blair. You're right. Maybe tomorrow we convince everybody else in the world.
You don't need to convince everyone else. You need to convince a couple people, and they need to convince a couple people, and I need to convince a couple of people. It's not a you problem, it's an us problem. And that's part of the issue here, is we can't be passing the buck around. We all have to do our own part.
We all can too.
Yeah. And we have to do both. We have to look at predictions, identify future threats, and work together to shield ourselves from those threats, to safeguard each other from harm. We also have to reduce our emissions to give us the opportunity to make sure that future is less bleak than it could be.
Well, when you are looking at a bleak future, sometimes it's good to dig into a fictional novel.
Oh,
definitely.
Let's get some mind candy in here. What do we got?
Mind candy. Okay, have either of you read any Jane Austen novels?
Yes.
Not my thing.
What?
He said, not my thing.
Not your thing. And you know what, Justin? Not my thing either. I tried and really was never able to get past like the first chapter in any Jane Austen novel. But
Mr. Jackson, you're not worried about this season's yields of the summer crops?
Yep. Nope. Anyway.
You know what? I think I got off on the wrong stop. I'm going to get back on the library train and go to a different section, I think.
But I like that older gentleman, even though he might be my cousin.
Bye-bye!
Well, there are many, many people who do love the Jane Austen novels. one favorite is Pride and Prejudice
Oh, yes.
with the
Yes.
Mr. Darcy. Mr. Darcy, yes.
He spurned me once again. I think I like him more.
Oh my goodness, you got some hidden.
Oh dear. Well, anyway, did you know that there is a mouse sexual pheromone named after Mr. Darcy?
I mean, that tracks.
It's called Darcin.
I love it.
It's called Darcin, yes. And a team of researchers at Zuckerman Institute at Columbia University wanted to look into what Darcin was doing in the mouse brain, specifically the female mouse brain. What they know is that when male mice they can urinate on things, leave a mark of their scent. Females will go and sniff it, and if it contains Darcin, these researchers published in Nature that this single protein, this little pheromonal protein, triggers a cascade within the brain that makes a female mouse excited, increase their sexual stimulated behaviors, and makes them sing little ultrasonic songs. But if these females happen to be lactating or pregnant, say they're moms and taking care of young already, it had no such effect. Meanwhile, they looked at the brain, right? They're like, oh, what's it doing? Okay, it gets in there, has this behavioral effect. What's going on in there? So they looked at the brain and they found that it goes from the olfactory bulb, stimulating an area of the amygdala, which seems odd because the amygdala is known to be associated with anger and fear, not necessarily mating responses. But they found, okay, these stimulated females, this part of their brain is excited. So let's go the other way around. And they took females and stimulated that part of their brain. And by artificially stimulating that part of the amygdala, they triggered those mating responses by the females. And when they shut off activity in that part of the brain, they could block the behavioral responses that Darcin would normally elicit. So they found this area of the amygdala is essential for this, and they think that it somehow mediates the internal state of the female. Hey, am I able to breed? Am I pregnant? What's going on in here? And it mediates that internal mental brain state, physiological state, with this outside stimulus to then lead to a behavior to mate or no behavioral response. But anyway, I thought it was really interesting.
Ooh, Kiki froze. It was very interesting.
It was very interesting. So it's kind of the thing I was just thinking is how logical the brain is without us.
That love and hate are in the same place? Is that what you're saying?
No. But I'm not picturing the female mouse debating the fact like, I know I've already got kids at home, but I really want to go clubbing. Mm-hmm. It's not. Yeah, that's a bad choice. It's not a moral decision or a logical decision process being made. The brain is making those logical decisions without you needing to contemplate. And I wonder how much, you think so much of humanity as having chosen. At some point, somebody came down from a mountain and said, okay, from now on, thou shalt not kill. Oh, never occurred to us. We've been running around just killing people like whenever we were in a bad mood up until this point. Right? No, that's not how it happened. The brain knew it's bad to kill all of your friends and family in your group because that's going to make it harder to survive. Because and it makes you actually, I love them. I know that my sibling treats me really badly. I don't know why I like them, but I love them, even though they're awful. And I probably should kill them. You know what? No.
Their
This is
success
a-
is the success of my genes.
It's a success of genes, but the thing is, it's the brain is taking care of so much logic that we've written it down. We've sort of transcribed like so much of human law and human the trappings of human society, or us just writing down what the brain has been making us do or inferring we should do for better survival from the beginning anyway.
You say it's the brain. I say it's hormones.
But what's reading
Fine
it? Like, yes,
interplay.
I agree. I was the whole brain hormone. Absolutely.
Yeah, no, they're
But
definitely working in concert. But
no, they're not working. The brain is what's sending those hormones. There's a control center in the brain that's like, yeah, more hormone. Order up some more hormone. We got this going on. There's a catalyst. Doesn't it run through the brain, or do you think it's just? I mean, I think it has to run through the brain to alter behavior. And yeah, the brain is looking for that hormone signal. It's its way of looking out into the world and seeing what's going on to the body, looking out into the body, seeing what's going on.
But in this case, it was a pheromone that caused all this to happen. Which is an
outside.
external. It's
Yeah.
an external source. So that's actually the brain responding to an outside stimuli. Which my point is, in a lot of cases, the brain's responding to something.
Mm-hmm.
It's the hormones that are in charge. The brain is just responding.
Okay, fair enough. But then it's also a natural selection that comes into play, right? So,
Of course.
for over generations, there were mice that were like, pheromone, who cares? I'm gonna go bad outcome. The young died, didn't work out well. They kept trying to make, but the ones that they did respond to it are the ones that have continued. So
Of
it's
course.
evolution.
Yeah, absolutely.
Absolutely.
I think a lot of it is hormones.
That's a lot of
It
things.
comes down to hormones. Kiki, are you eating cereal? Because you're snap, crackle, and popping.
And popping, yeah, right. Yeah. Oh my goodness, that was hilarious.
Thank you for that.
Oh, all
Better.
good. Yeah, fixed.
All right. Thank you. Okay. It's time to keep
Mm.
moving on this here.
Are we at the quick?
This story bus. Yeah, it's time for this bus to speed up and tell some quick stories.
All right, I got the
Wow.
quickest one.
You have the quickest
I
one?
got the quickest one. This is published in the open access journal EPJ Data Science. Basically, they went through and evaluated all sorts of classical romantic era classical music, 1700 to 1900, and ranked them based on complexity and innovation of the composers. Number one, first place, the most complicated, innovative composer, the most unique. It's sort of like when they take your school paper and they run it against the plagiarism filter. So a little bit of that going on here, right? Number one, most unique and innovative, Rachmaninoff. First place, not surprised. Second, Bach. Brahms, and then Mendelssohn. It was in order of the top there. Yeah.
Interesting.
Top. Blair, I have a hippo story for you.
Oh, give me
Yeah,
the
what
hippo
was it?
story. How did I miss it?
It's a
I don't know.
curious title too.
I don't know how you missed this story because it's not just hippos. It is the hippos of the drug lord Pablo Escobar.
Oh, yes. The South American hippos.
South American hippos. Yes. In
Wait.
Colombia.
What?
Yes.
Wait, what?
Yes.
They have
Okay.
meat.
Let me tell you this story.
South American hippo?
It is the new species, well, otherwise known as an invasive species at this point. When Pablo Escobar of Narcos fame, and anyway, when he was at his height, he had a lot of exotic animals in a family zoo. And when they took the empire down, they housed most of those exotic animals in other zoos or private collections or other. They found homes for most of them, but they didn't find homes for the hippos, and the hippos ended up in the local ecosystem. And so the hippos in Colombia are invasive hippos.
Mhm.
So what's interesting is the hippo. That's almost a megafauna.
It's definitely megafauna.
It's
That's
definitely a
gotta
megafauna.
That's
yeah.
a megafauna. That's gonna be changing an environment.
Oh
That's
yeah,
a
they're
mover.
keystone
Yeah.
species in Africa.
Yeah.
We've discussed. So
Yes, and that is the
Yeah.
point of this story. Researchers from UC San Diego have gone to Colombia to take a look at these hippos and study water quality, oxygen levels, isotope signatures, comparing lakes with hippos and without hippos, because the population grew from something like 4 hippos to now over 80
Oh
hippos.
wow, that's a lot of
And
hippos.
It's a lot of hippos, and they expect it won't be long before there are at least a couple thousand hippos in South America. And since they are keystone species, they want to know what the hippos are doing to the ecosystem. And it doesn't look good. In fact, the hippos are changing the water quality in the area. They are, because they go and they eat, and then they excrete those nutrients into the water.
which is why they're
And-
keystone species in Africa, because they're bringing nutrients into the water during a drought, but it is not quite so dry in Colombia.
Yes, and so it's changing the nutrient levels in the lakes. It is increasing increasing algae production. So with more algae, it can lead to algal blooms, and this is an interesting way to look at the effect of a keystone species on an ecosystem. And ecologists don't often have opportunities like this. So they are taking advantage of it while they can. But at the same time, they're hoping that they will be able to get information that can help the Colombian government figure out what to do with the hippos.
So
Because it's not easy to catch hippos,
if they don't decide to simply eliminate them,
which they could.
They could try.
if they don't,
it's gonna be tough.
Blair,
Yeah.
would you be in favor of introducing some other hippos to try to diversify that gene pool? If they decide not to kill them all, if they're going to allow there to be hippos as part of the environment, they're going to be like, we have Colombian hippos now. Would you be okay with importing an additional couple of hippos into the mix to diversify the gene pool of the hippos that they've decided they're going to allow?
So that's an interesting question. I was
Yeah.
about to mention that I feel like the opposite is likely to happen, and that is that if you let the population go far enough, if it only came from a couple, then there's a really good chance that some sort of disease or something is going to
Wipe them out.
wipe them all out,
Yes.
which actually would solve the problem.
But now, Blair, you've been hired by Colombia to be the curator of their hippo population, of their wild hippos. You're now in charge.
I appreciate what you're trying to say here, and I
Yeah.
love hippos, and you know
Yeah,
that, and that's
I
why
do.
you're pushing
That's-
here.
No,
But
it's
they
a perfect
don't-
job.
they don't belong there. And if
Yeah,
they're
that decision's
causing
already been made,
huge
Blair.
problems, no, if they're causing huge
Decision's
problems in the
already been made,
ecosystem,
Blair.
letting them be could actually make their population fizzle out.
Okay.
It's
There
possible.
you go.
It's absolutely possible. So this is the other problem, right? Is it's not just the poop, it's not just the water. Hippos flatten the earth and create these like flattened pathways.
I
when
didn't know they were
graze.
flat earthers. Now I'm for
which-
taking them
yeah,
out.
yeah.
I don't want them.
Yeah.
I don't
So,
want 'em.
but so if in a rainforest, that is bad because the understory is where there's this
Right.
huge amount of life. That's not good. There's oh my god, it's such a bad space for them. This is very reminiscent of the fact that we almost had hippos intentionally released into the United States in the bayou. That almost happened. So there's a fascinating book about it. There was an episode of maybe it was Science Friday or it was some NPR show way back in the day. I heard on the radio, but there was this whole thing where there was a campaign to release hippos in the United States because they thought that they would eat invasive water lilies. and they said that it would be cheap meat.
Hmm.
But no. Bad idea.
Yeah, nobody. I'm trying to like. Actually, I can actually picture it now. The domesticated hippo, and he's replacing the cow. So instead of these big open fields of cows up on the hill or something, you've got these large water features, which is hippos carousing around. Yeah, I can see it.
Yes.
Why didn't we? Hippos must not taste very good. That must be it.
They're pretty hard to kill. They're very territorial. They are dangerous. They don't want to be in a farm full of each other.
Hey.
That's not what they want. The story, the book
Is
called
better than
American
I.
Hippo, River of Teeth, Taste of Marrow.
Okay.
So it's a 2017 book. It's very interesting.
Sounds frightening. But you know what? The animal that kills more people in the United States than any other?
Well, mosquitoes.
Cows.
Oh.
Yeah.
It's cows.
You haven't heard him say that before?
Yeah, I
it's the
know,
most
but mosquitoes. I have one
Blair.
more story before the end of the show. My goodness. I'll just give you the liner notes here. Wasps are pretty smart. This is a study from Cornell University. It appears that paper wasps can recognize faces. Not human faces, but wasp faces. This is something that they have in common, most likely with other social insects that have multiple queens. In groups with a single queen, like honeybees, roles are clear, no need to tell each other apart. But paper wasps can have 5 queens, for example. Then they need to be able to tell which queen they're looking at. So they have capability for facial recognition. And so this is a huge jump in cognitive evolution between kind of similar types of animals that have totally different complex social structures. But yeah, paper wasps can tell faces.
I'm suddenly picturing a very small hive, 5 queens, 1 worker wasp.
Yeah.
Worker wasp, like, really? Everybody gets to be the queen with me?
It's good to be the queen.
I guess so. I guess I'm the only one that's got to go to do the work, though, right? Okay, I see how this works.
Yeah. But I know, I can tell. I know which of you told me to do which job.
Yeah, I got to keep track of 5 of you.
Oh, sure. Yeah, I did your laundry. Judith. I see you.
No, Judith is sweet. Judith is the good one. Judith is the nice one.
We have come to the end of our show. We have run through all of our stories for the night, and we hope that you have enjoyed them as much as we have enjoyed talking about them. I would love to say thank you for spending your time with us. Thank you to Fada for helping with show notes, social media, and also with our YouTube chat room. Thank you to Gord for monitoring our twist.org/live chat room. And thank you to Identity for recording the show. I also would like to thank our Patreon sponsors. Thank you to Paul Disney, Stu Pollock, Ed Dyer, Andrew Swanson, Craig Landon, Ed, Philip Shane, Ken Hayes, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Richard Porter, Mark Mazaros, Bob Calder, Jack, Matthew Litwin, Jason Roberts, Bill K., Eric Knapp, Richard, Brian Condren, Dave Neighbor, Ben Bignell, Justin Taylor PS, Josiah Zahner, Howard Tan, Donald Mundis, Sarah Forfar, Dan K., Matt Bass, Darwin Hannon, Patrick Pecoraro, Gene Tellier, John Gridley, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney Lewis, Tiffany Boyd, Mountain Sloth, Seth Gradney, Stephen Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Paul Ronovich, Sue Doster, Dave Wilkinson, Noodles, Kevin Reardon, Christoph Zuknarek, Ashish Pant, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Suzuki, Drim Drapeau, Greg Riley, Sean Lamb, Steve Leeson, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert Garrett, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hetzenflow, Kevin Parachan, Byron Lee, and E.O. Thank you for all of your support on Patreon. I would also like to thank our sponsor, the Burroughs Welcome Fund, for their generous support. If you would like to support This Week in Science, you can find information at twis.org, or you can go directly to patreon.com/thisweekinscience to help us keep bringing you this valuable science discussion on a weekly basis. On next week's show, we'll be back Wednesday, not Thursday, Wednesday at 8 p.m. Pacific time on twist.org/live. You can watch and join our chat room, but if you can't make it, don't worry, because it all gets saved to YouTube and to Facebook.
Yeah, thank you for enjoying the show. TWiS is also available as a podcast. Just Google This Week in Science wherever podcasts are found. And if you enjoyed the show, remember, tell your friends about TWiS.
For more information on anything you've heard here today, if you would like pictures of butterfly wings or SARS maps, show notes will be available on our website. That's at www.twist.org. While you're there, you can also make comments and start conversations with the hosts or other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in the subject line. Otherwise, your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover, address, a suggestion for an interview, a haiku that comes due in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy.
Like coronavirus.
This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science.
This week in science. This week in science. This week in science. This week in science. This week in science. This week in science.
TWIS.org. Go to TWIS.org. Subscribe to our newsletter, man. Yeah, got a newsletter. We might make a new one someday.
We definitely will.
We're in the after show, everyone. If you are watching and you are here, thank you for sticking around until the end of the show. So I keep wanting to add Justin back, but he's just a nothing person. He's just a person figure. Could potentially add him to the stream, but he's not there. So we have one more show next week, and then we go to AAAS for Valentine's weekend.
That'll be fun.
Yeah, and although we have a show scheduled for that Wednesday, I believe that will be a lot to ask.
Yeah, I don't know if that's going to be tough because I leave early on Thursday morning. I think you do too, right? So
Yeah.
That's going to be a lot.
Michael74 is saying if TWIS is 20 years old, that's older than TWIT. It is.
It is indeed.
I was TWIS before Leo was TWIT.
Mm-hmm.
It's true. I've been around too far. Noodles, yes, the next show is in February. It is. Ed wants a Mississippi hippo on the next calendar.
There's a hippo on.
I know,
Calendar
but was it a
before last.
Mississippi hippo?
Yeah, but that's just a hippo in a slightly greener backdrop. I could do a pygmy hippo on the next calendar. That's a different species.
Oh, they're cute.
Mm-hmm. I prefer Nile, but I'll take a pygmy hippo.
There is a woman who brought a hedgehog to my son's karate school today.
That's the kind of stuff I do.
I know. No, she's a mom, and apparently she went to a snake show and came home with a hedgehog. Interesting.
So Oregon is not a state where hedgehogs are illegal, then?
I guess not. Yeah. What's that smile for, Blair?
They are an extreme threat of becoming an invasive species, so that's why they're usually illegal. Because nothing in the United States knows how to eat a hedgehog, is the thing.
Yeah. No. You have to get to its soft underbelly.
Yeah. Which you have to sneak up on it.
Yeah, because they roll into little balls.
Does this look comfortable? This does not look comfortable to me. Like, how can you breathe?
I don't know. She looks happy. She looks very asleep.
Sleepy baby.
Sleepy Sadie. Yeah, Sleepy Sadie. Where's Justin? He's there, but not there. He's not even chatting at me. Did he just hang up?
I don't know. Michael asked me, Michael74 in the chat room asked what I did before, or what I do now, since I guess I called myself a former zookeeper recently. I haven't been a zookeeper in like 7 years. It's been a while. But I work in education now. So I run the Zoomobile. So we take animals into classrooms. I run some teen volunteer programs, and I do a lot of climate change communication. So I still work at a zoo. I still work with animals. Sometimes I put hedgehogs in carriers and take them to classrooms. That's a thing that I do. So, anyway.
Take hedgehogs to classrooms.
Yeah. I don't think Science Friday was a podcast before. I feel like that actually happened kind of recently.
No, they apparently... I don't know. I need to go back in the Wayback Machine. I looked in the Wayback Machine to find their podcast, and their podcast on their website wasn't there on their website until after ours
Mhm.
was already out, which is why I say we're the oldest science podcast.
Mhm.
Science Friday was definitely a
Yeah.
show
Yeah.
before we were, but I mean, they were around for a long time. I was inspired by Science Friday. Yeah, and we always seem to be within an episode or two from the Skeptic's Guide to the Universe. They started around the same time, but then at one point, I think they were doing more episodes in a week or something, or I took some vacations or something. I don't know. I mean, the episode number, I will say, this is my little inside joke on TWiS history. The number, there was a day when I realized maybe I should change the format of how I title the podcast. People like numbers. They like to know what number it is, because I had been only titling it by the date and not a number, because I wasn't counting what show I was on when we were just doing the radio show. And so then it was just whatever day it was, this is the show from that day. And so at a certain point, I went, oh, well, people like numbers in their podcast titles. So maybe we should put a number in our podcast title. And then I estimated, based on how long we had been podcasting, approximately how many episodes that would be. And I said, we're starting here.
That makes sense.
So the number of podcasts is not necessarily
It's approximate.
accurate.
It's approximate.
Uh-oh.
It's approximate. But if you go back and look through the records, there's a certain point at which the titling of the shows changed and we suddenly had numbers where there had been no numbers before. It's magic. And now you know. Now you know about the secret of the TWiS episode numbers. But really, we have been on the air since 2000, and we podcasting since 2005. And then Justin came on, and then the iTunes library, and then all the things. Yes.
I came
And
on
I'm one of those things.
I came
You're
on
one of the
right
things.
before podcasting.
Yeah, you did. You
The
started.
year of.
Mm-hmm. Yeah, you were a couple of episodes a little bit before we started the podcast.
If you took 52, which is the number of weeks in a year,
Yeah.
and
then
multiplied it by the number of years you've been on the air, it's over 1,000 for episodes.
Yes.
But yeah, for the podcast,
And I usually, I'm like, well, an episode here or there we didn't do, whatever. So I have always rounded it to 50.
yeah. I still think about the times when we did 2 episodes a week. It wasn't for that long, but we did it long enough, I think, to make up for a couple a year.
There was that, yeah.
We did it for so if it was 15. What episode does it say we're on now?
756.
If you just took the podcast years, it's 780.
We're higher number now.
We're probably. I think you probably underestimated by a little bit, but yeah, there's been more episodes than you can shake a stick at.
Yeah, it's true. Identity 4, I'm sorry, I have destroyed your whole world. The world is a lie.
Blair, there was somewhere in the comment feeds, I don't see it now, but somebody was asking why in the Blair's Animal Corner it says except for giant pandas and squirrels.
Yes, I did see that. And somebody did do a quick explanation of the panda side of things.
That you just hate pandas.
No,
Fine.
pandas, it all started, if I may, because pandas are like the poster child of endangered species. And so it's first of all, this like they're the they're the scapegoat for me of cute animals, this charismatic megafauna getting all of the conservation dollars, and animals that are perhaps not as cute or fluffy not getting as much financial support from the everyday person because of that. So there's I have my own chip on my shoulder about that to begin with. But then on top of that, we put a lot of effort into breeding pandas and trying to keep pandas alive, and they do everything in their power to not survive. And so that's very frustrating to me, is that they're bad at being pandas, and still we're just trying to hold their hands every step of the way, trying to get them to survive. It's a lost. It's the one species I'm convinced that would be extinct if it weren't for humans. They seem like an evolutionary dead end. Let them fizzle out. Anyway, that's the whole thing. The squirrels is just, I don't trust them. They're up to something, and it's never good. My experience with squirrels is they're very easily imprinted, and once they are, they're jerks. They're just so bad. They're so aggressive and bold, and so there was this. I never liked them very much, but also working at a zoo, that means that there are animals that are used to picking food out of the garbage and getting fed by humans. And so the squirrels at the zoo are very, very, very bold. And I was an intern at one point, and there was this squirrel that saw I was wearing keeper clothes and was getting closer and closer to me. One point put its paw on my foot, and I was like, get out of here. I shook my foot, and then it ran up behind me and jumped onto my leg. I'm jumping up and down one-legged with guests around. This is in public space.
I had not heard this story.
Trying to get this squirrel off me. Meanwhile, what's happening in my brain is, this squirrel is too bold. It's probably rabid. I'm about to get bit by a rabid squirrel.
Yeah.
When you get bit by something that's rabid, you don't want to move around. You don't want your heart to move too fast. That circulates it through your blood too fast. So I have to sit quietly down, call security, and have them call 911 if I get bit by this squirrel and it's rabid. So this is what's happening in my brain. Then I finally get it to run away. And I'm updating a board, a public notice board with a schedule of events, right? And so I'm trying to do this thing, and the squirrel won't leave me alone, and it jumps on my leg, and I get rid of it and everything. And then I hear this little rustling up over my head, and this squirrel is coming down the pillar towards my hand. And I drop everything I'm doing, and I go, that's it. And I walk away.
Oh my goodness.
I don't trust them.
They're out to get you.
I don't trust them.
Here's my idea though. So at some point we're gonna have, this has got, if I say it, it can't be a surprise anymore. But no, at some point we're gonna have a TWiS anniversary birthday gathering, number of episodes party, and when we do, I am going to invite people to wear giant panda and squirrel costumes.
Oh no.
And it'll be a whole party of giant pandas and squirrels.
I will say I received nothing but hippo items for a very long time, but now I get a lot of squirrel and panda items. A lot.
People know.
And at first I was frustrated by it, and now I think it's pretty funny.
It is hilarious. It's great. Where'd Justin go?
I've had people say, do you really like pandas? Are pandas your favorite animal? I just be like,
no.
No,
not
why
my
do you have so many panda
animal.
things? Don't worry about it.
Don't worry about the pandas.
This dog is fully asleep in my lap. How cute.
So I was in my lap. I was letting my 6-year-old drive, and this big empty parking lot in the back
Seems
said
safe.
campus.
Yep.
This is a tradition. I let the so the 6-year-old's on my lap. She's doing the little shifter and she's steering the car. And there's turkeys. I wish I had to keep monitoring the situation. Otherwise, I should have really grabbed the phone and filmed it. But these 2 turkeys, as we drove by, came running up to the car and ran one on either side of us as we drove through. And then we turned around and came back again. And they did the same thing. They like joined in and like ran alongside the car the whole time. And it was, I couldn't tell if they were like.
You were about to die. Have you heard of the episode? I talk about it all the time. The episode of This American Life. They usually play it every Thanksgiving.
Oh no.
And it was a person who was stuck in their house and then in a car by a tom turkey for like 3 days.
Oh no. Oh no.
Turkeys
are no joke.
So there was a very aggressive tom that got named Downtown Tom here in the city of Davis. That was menacing people, and of course it's Davis. So there's this big controversy about whether or not we should do anything about
the turkey alone.
The city of Davis finally decided we're getting rid of the turkey.
Good.
And I know the guy who had to take down Downtown Tom, and he was literally afraid of repercussion. like the town was going to turn on him because he got tasked with.
Did he eat him?
No, I don't know. I don't think so. I don't think that was part of it.
Waste not.
Yeah, you would think, but there are what are they called? Packs, herds, flocks of turkeys.
Probably
gaggles?
a flock. Probably
murder
a flock.
of turkeys.
No. Let me look.
A mischief? It's just mice.
A mischief of turkeys.
What is the term?
I'm looking it up.
For a group of turkeys.
Internet says, oh, these are fake. One of them says gobble. That's not right. You're incorrect.
Gobble.
Internet,
turkey.
you are incorrect.
I like it.
Rafter or flock?
A rafter. Yeah, that sounds right.
Rafter. This is the traditional one. Rafter.
So there's different rafters of turkeys. And amazingly, we talk about animal intelligence, and it just could be a coincidence. I don't know. I'm at the intersection, and there's a big rafter of turkeys hanging out at one side of the intersection. And they're just all sort of gathering there. The light for them turns green. The little crosswalk walk turns, and they walk through the crosswalk during during their green, and made it across. And I'm thinking, I've seen them wandering in the middle of the street in the suburb parts of town, but this was like the busier intersection with the light and everything. And they waited for the green and went across in the crosswalk. Mm-hmm. Yeah, they're smarter than you think. Maybe.
There you go.
Maybe.
But they're phenomenally ugly looking up close.
Don't say that.
They are. They've got really neat colorings of red and blue, but then they've got all this like almost toad-like bumps all over, and this extra like this extra like neck feather thing.
Yeah, the wattle.
No, not just like the wattle here, but like a feather, like a chest feather.
Oh, yeah.
That was just sticking out for no apparent reason. Yeah, and they make weird sounds. The toms make this weird.
Yeah, totally.
Oh, they're loud. They're weird. Almost be where it was the first. I think Ben Franklin wanted to make it the national bird.
Yeah, he did.
Yeah.
Because he didn't like how eagles were scavengers. He thought they were thieves.
Interesting. Also, eagles don't really sound that impressive.
No, they don't. They sound like they're dying.
Yeah, the sound that the sound that people associate with an eagle is actually I think some kind of red-tailed hawk or something.
Yes.
That-
Yeah, it's a
that-
red-tailed hawk.
That's a red-tailed hawk.
Ah! Yeah, that's a red-tailed hawk.
Like that's the hawk sound.
Yeah.
The eagle sounds nothing like this.
Yeah. I think they have used eagles for various dinosaurs in the past, though. Because they do sound kind of dumb.
It was before you joined the show, but I should do this again. I took chickens and I slowed them down.
Oh, yeah,
with, like, a
yeah.
pitch
That's the
shifter and a slow-
T-Rex.
And it just, that's the sound. It was like perfect T-Rex sound. It's just,
The other sound they use for dinosaurs all the time is elephant seals.
Interesting.
Sorry, I'm looking at old pages of like This Week in Science links on the Wayback Machine.
Oh,
Yeah.
interesting.
I don't know where things are. They're all these M3U links. M3U? What was I doing? What was I thinking?
This dog is almost too long to fit on my lap. What will I do then?
I wonder how far back I do have things. Interesting.
You're almost too long. Look at how long this dog is.
That's a long dog.
You're so
Extra long dog. I went to a cat show this last weekend.
You did?
I did. And we got to pet all the cats. And it was wonderful. I was thinking that we'd go to the cat show and everybody would be like, don't touch my cat. My cat is special. Don't touch my cat. But it was all about touching the cats, petting the cats. And we got
That's so fun.
it was really fun. We got to play with all these cats and they were all like, I guess part of the judging of cats has to do with their personalities. They must be playful. They must be personable and playful.
It's a rare cat.
I had no idea. But something, especially because I was paying attention to the nCoV news all weekend, I'm like, constantly on Twitter trying to figure out what was going on. I was there and realized that we kept having to put hand sanitizer on our hands to be able to touch people's cats. They were like having us put hand sanitize so that we weren't spreading disease between the cats and also from person to cats. But the idea is that, okay, got to protect the cats. And I realized it was probably one of the safest places to be for some kind of in the cold and flu season. I'm like, everybody's cleaning their hands the whole time. Everybody's going to be fine.
Yeah.
Sure. Oh, that puppy. Now she's like, I don't know what to do. I'm tired, but I have to pee. What's happening?
I'm tired too.
Oh,
Sadie. Yeah.
Yes, I probably already have toxo though, Ed. You all know that. I love my
I know, and I've spent many years picking up otter poop, so I might also have toxo.
No.
No.
Who's to say?
Who's to say? Who knows?
Well, I might have gotten toxo from the lions and tigers too.
I have suggested before as a fun aside episode, we could all go get tested.
Yeah.
I know, we could.
Actually, we could. That would be fine.
I don't want to know this.
I don't know why I know this until after I have children.
Doing just fine.
Why wouldn't you want to know?
Well, for me, I feel like if I find out I'm carrying it, I probably shouldn't have children, right?
No.
No.
Well,
I think it's
not
just
right
to be getting
then.
fit. Yeah. Well,
Yeah, get treated. That's why you want to
treated.
know.
Oh, yeah.
Exactly why you'd want to know. Especially now.
Good night, Noodles.
Go get him.
Oh, Justin has to be in the same room with us in Seattle. Mwahaha.
Huh?
Thunder Beaver is saying that with all the toxo between me and Blair, you probably never want to be in the same room with us again.
Beware. Yeah.
Justin, beware. Yeah, Seattle. That's right. We will be there in 2 weeks. And Identity, I think you said something about wanting to know when we were going to do a meetup. We do not have a specific time for a meetup
No,
at this point in time. What?
not yet.
No, I know. Not
don't.
yet. I'm
We
waiting to
will.
hear. I'm waiting to hear. There's supposed to be for the podcasters. There's supposed to be a get together, and I was thinking it would be. I don't know. We could do a crossover. I don't know. I don't know when and where. There's a lot of events. Are our TWiS shows open to the public at all? No. Which is really unfortunate. There is the family days on Saturday, and our show is at 11:30 on Saturday. But it's not in the expo hall where it's in the expo hall for the conference, but not where the family days is going to be. And I don't know if you can get into that as a family days attendee. It is a bummer. It is a bummer, but it would be nice to be able to connect IRL. I like that. If we're into town, we should hang out. It would be great.
I'm for it.
Yeah, I'm for it also. But I will, I mean, Friday would probably be the best day, I imagine. I mean, there's other things as well, but Friday might be the best day. Or for me, yeah, I don't know. I got to look at the schedule again. There's a lot going on. I think Friday might be best. We're quarantined. No.
I like being called a science wizard, though. Keep that up, Identity 4.
Yeah. I'm a science wizard.
That's great. I love that.
I will cast magic missile. Yeah. Gaurav Sharma said turkeys is a rafter. Yep, also.
Mm-hmm.
I am tired. You're tired. I think it is time for us to go. Do we have anything else that we want to talk about before Justin says peace out? Anything to remember?
We forgot to say good night, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin.
Good night,
Kiki.
Kiki.
Good night, everyone. Thank you for being a part of this show once again on Wednesday, not Thursday. We'll be back again next week. I hope you sleep well and have happy science dreams. We will see you again next week. Good night.